In September 2021, a company was founded in Dallas, Texas, with a stated goal: bring back the woolly mammoth. It is called Colossal Biosciences, and was founded by the Harvard geneticist George Church and the entrepreneur Ben Lamb. In 2023, the company stated it wanted woolly mammoth hybrid calves by 2028 and wanted to reintroduce them to the Arctic tundra. The money followed: $15 million in seed funding, then 60, then 150, then 200 million more. By January 2025, Colossal had raised $435 million and was valued at $10.2 billion, the first company of that scale headquartered in Texas. So, is a mammoth coming back? The honest answer is more interesting than yes or no, and it turns on a question that sounds pedantic and is not: what would you actually be looking at? Because you cannot clone a mammoth, not because the technology is immature. Because of chemistry. Start with what was lost. The woolly mammoth began to diverge from the steppe mammoth about 800,000 years ago in Siberia. It was roughly the size of a modern African elephant. Males reached shoulder heights between about 2.7 and 3.5 meters and weighed between 4 and 8 tons. It had an outer coat of long guard hairs over a shorter undercoat in colors from dark to light. Its ears and tail were short to minimize frostbite and heat loss. It had 4 molars at a time, replaced 6 times across a life that could reach 60 years. A newborn calf weighed about 90 kilograms. Its home was the Mammoth Steppe, a cold, dry, grassy biome that stretched across northern Eurasia and North America, and no longer exists anywhere on Earth. Humans lived alongside them, hunted them, and used their bones and tusks for art, tools, and dwellings. And here's the fact that reframes the whole subject. Mainland Siberian mammoths persisted until about 10,000 years ago. On St. Paul Island until 5,600. On Wrangel Island in the Arctic Ocean until about 4,000 years ago. 4,000 years ago. Mammoths were alive on Wrangel Island, while cities stood in Mesopotamia and the pyramids were already old. They are not a lost world. They are recent. And there is a reason mammoths sit at the center of this subject that has nothing to do with genetics. Mammoths are the animal that taught us extinction was possible at all. Their remains were known in Siberia and North America long before European science looked at them, and were explained in a great many ways. The first remains studied by scientists were examined by the British physician Hans Sloane in 1728, fossilized teeth and tusks from Siberia. Sloane was the first to recognize that they belonged to elephants, which created a problem, because elephants live in the tropics. Sloane's explanation was the Great Flood. The animals had been buried during it, and Siberia had previously been tropical. Others preferred a variation, that the flood had carried elephants from the tropics to the Arctic. In 1738, the German zoologist Johann Philipp Brain argued the fossils were some kind of elephant, and could not explain the cold either. Nobody was being stupid. There was simply no available idea that a kind of animal could stop existing. In 1796, the French biologist Georges Cuvier identified the woolly mammoth not as a displaced modern elephant, but as an entirely new species, one that had gone extinct and no longer existed anywhere. That concept was not widely accepted at the time. It is the foundation of everything that follows. In this episode, extinction had to be invented as an idea before anyone could think about reversing it. Recent, and still not recoverable in the way films have taught us to expect. DNA is a molecule, and molecules break down. Ancient DNA degrades 3 ways. It fragments into shorter and shorter pieces, it cross-links, and it deaminates. Individual bases chemically change into other bases. That last one is so reliable that it is used as a test of authenticity. Cytosine decays to uracil at the ends of fragments and reads back as thymine. C to T substitutions, increasing with age, are the fingerprint of genuinely ancient DNA. Even under the best possible preservation, there is an upper boundary of roughly 400,000 to 1.5 million years for a sample to hold enough DNA to sequence. Studies modeling decay in moa bones found that mitochondrial DNA would degrade to an average length of 1 base pair after about 6.8 million years at -5 degrees. 1 base pair, which means dinosaur DNA, 66 million years old at the youngest, is not a technology problem. It is thermodynamically gone. In the 1990s, this was genuinely unclear. Papers reported DNA from insects in amber, from a Cretaceous egg, from dinosaur bones more than 80 million years old. Every one of those claims was later dismissed. The reported dinosaur DNA turned out to be human Y chromosome, contamination from the people doing the work. The field grew up around that failure. Extractions now happen under extreme sterile conditions, with molecular tags to distinguish genuine sample molecules from anything introduced during analysis. And once it grew up, it delivered. The oldest DNA sequence from a physical specimen comes from mammoth molars in Siberia, over 1 million years old. In 2022, 2 million-year-old genetic material was recovered from sediments in Greenland, currently the oldest DNA known. And that same year, Svante Pääbo won the Nobel Prize in Physiology or Medicine for his work on the genomes of extinct hominins. So mammoth DNA is real, abundant, and readable. What does not exist is a living mammoth cell. Cloning, the Dolly the sheep method, needs an intact nucleus from a living cell, transplanted into an egg of the nearest living relative. Cloning is the only method that would give you an animal with the original species genome. It is also the only method that is completely unavailable here because it needs a viable cell, and 40,000 years of freezing does not preserve one. So, the actual plan is different. You take the nearest living relative, the Asian elephant, and you edit it. The mammoth and the Asian elephant share about 99.6% of their DNA. Colossal has assembled an ancient mammoth genome using 65 mammoth genomes spanning about 700,000 years. Church's lab identified around 60 genes it hypothesized were responsible for the mammoth's distinctive traits, the high-domed skull, holding oxygen at low temperatures, fatty tissue. By 2017, the lab reported it had successfully added 45 genes to the genome of an Asian elephant. The target traits are specific and physical: a 10-centimeter layer of insulating fat, 5 different types of shaggy hair, and smaller ears, the things that let a big gray animal survive an Arctic winter. And that is the crux. Genome editing does not resurrect a species. It produces a proxy, a hybrid carrying selected traits of the extinct animal on the genetic background of the living one. Nobody serious disputes this, including the people building it. Which raises an obvious question: has anything ever actually been brought back? And the answer is yes, several times. Look closely at what they were. The Judean date palm was a cultivar native to the historical region of Judea, believed to have died out around the 15th century. Seeds recovered in the 1960s from excavations at Herod the Great's palace were given, in 2005, to Sarah Salin, who proposed germinating them. Her colleague Elaine Soloway revived several of them using hydration with a household baby bottle warmer, plus standard fertilizer and growth hormones. The first plant to grow was named Methuselah. By 2015, it was producing viable pollen. And in June 2021, a female plant named Hannah produced the first dates from that lineage in millennia. In 2010, Salon germinated another seed recovered in 1986 from a cave in the northern Judean desert. The resulting plant, named Shiva, reached maturity in 2024 and is believed to represent an entirely new species. The York groundsel was a small yellow-flowered plant first identified in York, England in 1979, last seen in the wild in 1991, and confirmed extinct by 2000, weed killer a contributing factor. Seeds preserved in the Millennium Seed Bank were germinated, and the plant was returned to York in 2023. That is the first documented case of an extinct species being revived and returned to its native habitat. The Montreal melon vanished from farms by the 1920s. In 1996, seeds turned up in a seed bank in Iowa, and local gardeners grew it again in its historical growing area. Notice the pattern. Every unambiguous de-extinction so far is a plant, revived from a seed that never stopped being alive. The nearest animal equivalent is the Florana giant tortoise, extinct on its island by around 1850. In 2012, hybrids carrying its ancestry were found on another island, and a breeding program launched in 2017 has hatched more than 400 tortoises. Which is not cloning and not gene editing. It is backbreeding, selecting, generation after generation for ancestral traits that still exist in a living population. And nature does a version of this without us. It is called iterative evolution. On the island of Aldabra, there was a flightless bird called the Aldabra rail. It went extinct about 136,000 years ago when rising sea levels drowned the island. About 100,000 years ago, the sea fell, the island came back, and the flying, white-throated rail recolonized it and evolved again into a flightless species physically identical to the extinct one. Same island, same starting bird, same answer. Given the right conditions, evolution will repeat itself. It just will not do it on a schedule. In April 2025, Colossal announced 3 gene-edited wolf pups, 2 males named Romulus and Remus, and a female named Khaleesi. The company presented them as dire wolves. The dire wolf genome had been read from 2 ancient samples, a 13,000-year-old tooth and a 72,000-year-old ear bone. Scientists compared gray wolf and dire wolf genomes, then rewrote 14 genes in gray wolf cells to express 20 traits they identified with the dire wolf's appearance. 45 engineered ova became embryos, and the embryos were carried by 2 surrogate hound mixes. Here is the part worth being exact about. no ancient dire wolf DNA was spliced into the gray wolf genome. The edits were written from scratch, guided by the ancient sequence. Independent experts disagreed sharply with the framing. One assessment was that the animals are not a dire wolf under any definition of a species ever. The International Union for Conservation of Nature's Canid Specialist Group formally declared the animals neither dire wolves nor proxies of dire wolves, judged against the Union's own guiding principles for creating proxies of extinct species. Their reasoning was ecological as much as taxonomic. Creating a phenotypic look-alike does not change the conservation status of an extinct species. There is no ecological niche left for it, and it will not restore ecosystem function. They concluded that the project does not contribute to conservation. Colossal published a response arguing its project does align with those principles. And then in May 2025, the company's own chief scientist, Beth Shapiro, described the three animals as gray wolves with 20 edits, said that had been the company's position from the start, and acknowledged that it is impossible to bring back an organism identical to a species that used to be alive. The term dire wolves, she said, is a colloquialism, not scientific terminology. That is the clearest statement anyone has made about this entire field, and it came from inside the company doing the work. A similar argument attended the woolly mice, laboratory mice given mammoth-inspired mutations in March 2025, with long, shaggy, tawny fur. Some experts argued the experiment was really about mouse genetics. Suppose all the editing works. You still have to gestate the animal. The plan is to use African and Asian elephants as surrogates, and to develop artificial elephant wombs lined with uterine tissue as a parallel path. That is not a small ask. Asian elephants are endangered, and an elephant pregnancy runs to nearly 2 years, which brings the strongest criticism of de-extinction, and it is a resource argument. Bringing back one species can cost millions, and that money most plausibly comes out of conservation. Critically endangered species could go extinct faster if funding shifts away from keeping them alive. There are ecological risks too. A revived species may find its niche already filled, and behave as an invasive. It may lack immunity to diseases now present. It may simply be hunted again. A mammoth carries ivory, and ivory is why elephants are poached now. And a proxy that does not behave like the original may not do the ecological job the original did, which is usually the stated reason for making it. Now the other side honestly stated because the technology is producing real things, whether or not a mammoth ever walks. In 2022, Colossal and the Vertebrate Genomes Project sequenced the entire Asian elephant genome and released elephant genomes publicly for research without use restrictions. In October 2024, the company reported rebuilding a 99.9% accurate thylacine genome from a 110-year-old preserved specimen skull, described as the most complete ancient genome of any species known. In January 2025, they completed the thylacine genome and used a prototype artificial uterus to culture fertilized marsupial embryos past halfway through pregnancy. The company is developing a vaccine with Baylor College of Medicine for elephant endotheliotropic herpes virus, a virus that infects and kills many young Asian elephants. It is partnered with BioRescue on the northern white rhinoceros, which is functionally extinct. 2 females left, neither able to reproduce. And it banks tissue, the Sumatran rhinoceros, the red wolf, the vaquita, the northern quoll, the pink pigeon, the ivory-billed woodpecker. Whatever you think about mammoths, a functioning artificial marsupial uterus and a frozen library of endangered genomes are useful objects. The project list has kept expanding. The dodo, with the Nicobar pigeon as the closest living relative. The moa, with Peter Jackson and Canterbury Museum and Nai Tahu, and in April 2026, the bluebuck, extinct 2 centuries, the company's first project on mainland Africa. Not all of that has been welcomed. New Zealand scientists largely dismissed the moa project as unrealistic. One zoologist said it had nothing much to do with solving the global extinction crisis, and more to do with generating fundraising media coverage. Colossal rejected that characterization. Another researcher raised the question of consent, that iwi he had consulted were widely opposed to de-extincting moa, and wanted bone samples, DNA extracts, and sequence data to remain in New Zealand. That is a real question that genomics keeps running into. Whose material is it, and who decides? So, 2028, the stated target. What arrives, if it arrives? Most plausibly, an Asian elephant with a set of engineered cold adaptation traits, more hair, more fat, smaller ears. An animal that has never existed before, standing in a biome that no longer exists either. It would not be a mammoth, it would be something new, made deliberately out of one living species and the readable remains of a dead one. And there is a version of that outcome which is genuinely magnificent, provided nobody has to pretend it is a resurrection to make it worth doing. Extinction, as far as anyone can currently demonstrate, is still permanent. What is changing is our ability to read what was lost, and to write parts of it back into something living. Those are not the same thing. But the second one is not nothing.